Hello, everyone. Welcome to this lecture on lungs and heart. Let's go ahead and get started. I am going to share my screen now. All right. So here are the objectives we're going to cover. We'll start with the lungs. We'll talk about the airway with particular attention to changes from the upper airway to the lower airway. We'll compare and contrast major surface features of the right and left lungs, including the hilum of each lung. We'll describe structural relationships of the parietal and visceral pleura, explain the mechanism of pulmonary ventilation, and then we'll go into the heart. So we'll describe the anatomical position of the heart and its major vessels. We'll describe the layers and functions of the pericardium. We'll talk about the structure and function of each chamber and valve. Describe and diagram the cardiac cycle and how it relates to blood flow through the chambers, valves, and coronary vessels. We will describe the location of the major coronary vessels and their branches. And then we're going to put some concepts together. We'll talk briefly about auscultation of the chest wall and for lung sounds, and the auscultation of the chest wall for heart sounds. So let's start with the airway. Okay, so the airway is divided into two sections, the upper respiratory tract and the lower respiratory tract. Now, I actually consider the larynx, which is shown here, as part of the upper respiratory tract, and that's certainly arguable. The reason why I consider it part of the upper respiratory tract is because I consider it the gateway into the lower respiratory tract. But for our purposes, it really doesn't matter, just as long as you understand that the larynx is between the pharynx and the trachea. So air travels into the upper respiratory tract, typically via the nasal cavity, through the nares of the nose, and then through the nasal cavity where it is exposed to the warm epithelium, the warm respiratory epithelium located in that region. That respiratory epithelium does a number of things that you'll learn in histology. One of the things that it does is it traps particulate matter. It warms the air, as I mentioned, and it is one of the first sites of immunological response to any of the particulate matter that enters into the airway. In fact, we have tonsils in the back of the pharynx, and those are important for that immunological response. Airway then travels from the nasal cavity into the nasopharynx. The pharynx is a long tube that connects the nasal cavity, the oral cavity, nasal cavity, oral cavity, and then it transitions behind the larynx into the esophagus. So as air moves from the nasopharynx inferiorly through the oropharynx, it then enters into the larynx. Now, air, as you know, can also travel through the oral cavity and go into the oropharynx and into the larynx. What's important here is that air goes into the larynx and not into the last part or the most inferior part of the pharynx, which is called the laryngopharynx and clinically referred to as the hypopharynx. So air passes into the larynx through what's called the laryngeal inlet, and then through the rhimaglottitis, which is bordered by the vocal folds. It goes into the trachea, and then at a structure called the carina, which is where the trachea bifurcates, air can travel into the right mainstem bronchus or the left mainstem bronchus. The right mainstem bronchus, as you note here, is a little bit more vertical than the left mainstem bronchus, and that is because the heart sits here just to the left side of the thoracic cavity. So that has some clinical consequences as particulate matter or infection or foreign objects can tend to go into the right lung first simply because of gravity if the person has aspirated anything while they are standing up or sitting up. The other consequence of that also is if we need to drain the lungs. Draining the right lung is actually a bit easier than draining the left lung, because anything that needs to go out of the left lung needs to make this right angle turn. And that is certainly relevant in situations of cystic fibrosis. From the main stem bronchi, the airway branches many times into secondary bronchi and then tertiary bronchi before reaching eventually the alveoli. So here's the trachea in a little bit more detail. One thing to note is the C-shaped tracheal cartilages. These are really important to keep the airway patent or open. And the analogy I like to think about is a dryer vent, which has those aluminum ridges around the vent to keep it open. Notice that the trachea is preceded by, as far as cartilages go, the thyroid cartilage, which sits superior to it, and the cricoid cartilage. So thyroid cartilage and cricoid cartilage. Here again is the main stem right main stem bronchus. Here is the left main stem bronchus, which branched from the trachea at the carina. The epithelium of the trachea is a pseudostratified ciliated epithelium, also known as a respiratory epithelium. And the cilia are super important for being able to brush back particulate matter that might have gotten into the airway, things that snuck through the laryngeal inlet and the rhinoglottitis. And that material goes into the laryngopharynx and then is typically swallowed. Notice that we also have goblet cells in that epithelium, and those goblet cells are important for producing a mucus, which helps trap that particulate matter as well, and enables the cilia to move a mass movement of material towards the pharynx. One of the things to note also is that the posterior wall of the trachea is not solid, but it's a muscular structure. So this trachealis muscle has the ability to somewhat close the airway, but it's really more to provide stabilization and allow the cartilages to be taut without expanding too much. So the bronchial tree, as I mentioned before, includes primary bronchi, secondary bronchi, and tertiary bronchi. Here's the main stem or primary bronchus on the right side. This would be a secondary bronchus. And then further distal to that would be the tertiary bronchus. There's many bronchial divisions. The image on the bottom shows the transition from a main stem bronchus, and this is drawn linearly, by the way, from the main stem bronchus to a secondary bronchus to a tertiary bronchus. One of the things to note as we branch from section to section to section to section is that the tracheal, or it's not the tracheal cartilage, but the cartilage in general dissipates with every succeeding branch until we get to a region where there is no more cartilage surrounding the air tube. When we get to that point, the tube is called a bronchiole. It's microscopic in size. It no longer contains the cartilage, and it's surrounded by smooth muscle. And this smooth muscle has a clinical consequence, as this muscle is controlled by the autonomic nervous system and will close under parasympathetic control or open under sympathetic control. And there are, of course, bronchodilators that simulate the sympathetic nervous system to be able to open that airway. People with airway conditions that involve the bronchioles understand what that's like. And we'll look at an example of that in a little bit. I only put up the bronchopulmonary segments here just to illustrate that the tertiary bronchi are destined to deliver air to specific segments of the lung. I am not concerned that you know which segment is where, just that you understand that the lung is divided into functional segments. So here is a more detailed picture of the bronchiole as it's surrounded by smooth muscle. You'll notice that as the smooth muscle dissipates now, we now have an increasing number of alveoli. When the bronchiole located here transitions to a section where we have these outpocketings of alveoli. That's called the respiratory bronchiole. And eventually that respiratory bronchiole transitions into what's called the alveolar duct, which is shown here. And now we have pretty much only alveoli, but they are arranged in a straight line, and that gives way to the alveoli. So here is an alveolar duct that's shown in cross section. You could just as easily have a cross section located here, and it would still be the alveolar duct. But once we get into the grape-like structure, it's now the alveolar sacs. Alveoli, by the way, means grapes, or refers to a grape-like structure, and you can see that morphology here. Okay, so going back to this idea of smooth muscle in the bronchioles, condition of asthma is when the smooth muscles contract to narrow the airway. In addition to that, the epithelium located in the bronchiole is producing more mucus than usual. And so you have the double whammy of a constricted airway plus mucus. So bronchodilators help relax the smooth muscle, and that allows a person to cough the mucus out. Asthma is caused by many different things. Some common things would be pollen, smoke, mold, dust mites, air pollution, pet dander, exercise, or even anxiety. So that is the anatomical mechanism of asthma. At the alveolus, if I took a cross section of any one of these little air pockets, you would see something that looks like this. And what's important to note here is that this circular structure is the cross section of a sphere, and it forms what's called the respiratory membrane. The respiratory membrane is the location where type 1 alveolar cells form its wall, and then, type 1 alveolar cells form its wall. Oh, they're just labeled twice here. And then we have type 2 alveolar cells, which produce surfactant, also located along the wall. This wall is also surrounded by many capillaries, and this is where gas exchange is going to occur. And then lastly, the location of macrophages is all along the wall. The macrophages I like to think of as little rock climbers. They hold on to the wall. Sometimes they're squeezing through the wall. But these macrophages, also known as dust cells in the respiratory system, are the cells that are engulfing particulate matter that somehow made it past all of the defense mechanisms from the upper airway into the lower airway. For example, these dust cells are the cells that are engulfing tar from cigarette smoke. And when they engulf something, they will hold onto it and just stay attached to the respiratory membrane. If there is too much of an accumulation of particulate matter, these macrophages will actually migrate into the respiratory membrane and cause an inflammatory reaction. And it's that inflammatory reaction which can cause all sorts of problems, one of which could be eventual scarring of the respiratory membrane and actually destruction of the alveolar wall. Let's go back to the blood flow here. The capillaries are the terminal ends of the pulmonary artery and the beginnings of the pulmonary veins. So the pulmonary artery comes from the heart. It's delivering relatively deoxygenated blood to these alveolar sacs. We have an oxygen exchange in the sacs, taking up or getting rid of carbon dioxide and taking up oxygen. And then the oxygenated blood returns back to the heart via the pulmonary veins. Emphysema is a condition in which the respiratory membrane dies, and the effect of that is to transition pathologically from healthy lung tissue, which looks like this honeycomb shape, to a tissue in which the alveoli are destroyed, and you're left with these massive holes in the lung. And that causes all sorts of different problems, one of which is the recoil mechanism that helps air move out of the lungs is destroyed, which ends up trapping air into the lungs. And then, of course, if we have diminished numbers of respiratory membranes, then the gas exchange is also compromised. Okay, so now we're going to take a look at some lung features. One of the things to note about the lung features on the medial side, which is located down here at the bottom, is that these are features that are caused by the fixation of the lung during perfusion. These features don't actually exist on an in vivo live lung. So these are the relative locations of structures that are associated with the medial side of the lung. The lobes, however, are real, and so we'll talk about those now. The lung is a pyramidal structure with an apex located superiorly and a base located inferiorly. The right lung, shown here on the left, has 3 lobes. It has a superior lobe, a middle lobe, and an inferior lobe. And these are divided by an oblique fissure, which runs diagonally from superior to inferior, posterior to anterior. And then a horizontal fissure, which courses approximately horizontally to the rest of the lungs. And so that demarcates your superior lobe from your middle lobe from your inferior. The left lung, in contrast, only has 1 fissure. It's also called an oblique fissure, which results in a superior lobe and an inferior lobe. Okay, if we turn our attention now to the medial surface of the lung, there are 2 areas or 2 nomenclatures you should be aware of. The hilum is the region of the lung located here in which the root travels in and out of the lung. The root is comprised of the structures or the tubes that go in and out of the lung. That includes the pulmonary arteries, shown here in blue or purple, looks like blue, the bronchi, shown here in more of a white gray, and the veins, shown here in a red color. Remember that the pulmonary arteries are carrying deoxygenated blood and the pulmonary veins are carrying oxygenated blood. So something to note here on the right lung. The first is that the right main stem bronchus in this cut, in this sagittal cut, appears to be more like an oval structure. Whereas on the left side, it's a little bit more round, and it's not drawn quite that way here, but it's when you look at the cadavers, you'll note that the left main stem bronchus has more of a circular profile. And that is because of the way the bronchi are oriented in the thoracic cavity. So if I do a sagittal cut of a tube that's a little bit more vertical, then I'm going to get an oval profile from that cut. If I cut a tube that's a little bit more horizontal using a sagittal cut, I'm going to get more of a circular profile. So that's where that comes from. What's important to understand anatomically is that on the right side, the pulmonary artery is anterior to the primary bronchus. But on the left side, the pulmonary artery is superior to the left main stem bronchus. Basically everywhere else, any other vessel that you'll see in the hilum is a pulmonary vein. But one way to really verify that that's the case is to look at the thickness of the wall. The bronchus is going to have a very thick cartilaginous wall. The artery is going to have a thick muscular wall, but relatively less thick than the bronchus. And the pulmonary veins are going to have a thin muscular wall. So from thick to thin, bronchus, artery, vein. Okay, let's talk about the pleura. The concept of pleura and pericardium and serosa are the same. And the idea here is that the body is essentially a tube within a tube. And the cavities are continuous with what I would call the outside world. And if you review your embryological lectures, you'll note that the midline of the body closes, trapping part of the outside world inside the body and closing it off from the outside world, which is actually outside the body. So this outside world in the body, or this space, is called the body cavities, and those body cavities are thoracic, abdominal, and pelvic. Well, it turns out that the lungs and the heart develop into the thoracic cavity, or this space within my body. And so that has a consequence. The wall, the actual wall of my body, the thoracic wall, is a somatic structure, or I would say an inside world structure. And it does not want to be exposed to the outside world. The argument is the same if I am talking about my skin. My skin on my hand protects the skeletal muscular structures of my hand from the outside world out here. So I have a layer of the skin which has an epithelium, in this case an epidermis, that has an epithelium to protect me from the outside world. So if I look at this thoracic wall, we're going to look at the inset first. If I have in my thoracic wall, this side of the thoracic wall, shown by my arrow, will have skin on it. It has an epidermis protecting it from the outside world, which is located here to the right of the image. Then my inside world, or my body, consists of my musculoskeletal structures, at least in this image, and that includes the intercostal muscles and the ribs, and so it forms the wall. Well, if the outside world also exists in the cavity, then I need to protect my wall from that outside world. So I actually have a layer of tissue. It's also an epithelium, and in this case, it's a serosa. Now, the serosa is named differently from cavity to cavity to cavity. In the thoracic cavity, where the lungs are located, we call that serosa a pleura. So this is a pleural layer. It is a single-celled, simple squamous epithelium. All right, so that's a pleura. Now, we also have to protect the lungs, which are also inside world, and they develop in this cavity as well. So the lung needs a layer as well, a protective layer, and it will also be surrounded by a serosa. And so we call that layer a pleura as well. Well, we can't have two pleuras, and that doesn't make any sense, anatomically at least. So we give a special name to the different pleura or different serosa. The serosal layer, or the pleura, in in this part of the thoracic cavity that is surrounding the lung, but adjacent to the body wall, is called a parietal pleura. The serosa, or pleura, on the lung itself is called a visceral pleura. And so everything by definition, as far as serosas go, that are on an organ is going to be called visceral, and on the body wall called parietal. And that concept remains the same, whether we're talking about the lungs, which would have, again, a parietal pleura and a visceral pleura, or the heart, which would have a parietal pericardium or a visceral pericardium, or the gut, which would have a parietal peritoneum or a visceral peritoneum in terms of the concepts. Same type of tissue layer, different names based on the region. Okay, so where did all that outside world space go, that cavity that's filled with air? Well, that, by virtue of the lungs expanding into the pleural cavity, we have a little space here between the two layers of pleura. Now here it's labeled as pleural cavity. It is the pleural cavity, but when we talk about the pleural layers that are next to each other here, a more appropriate name for this is actually the pleural space. And so we have a serosal fluid produced by the parietal pleura and the visceral pleura, which fill in this pleural space. And it's that fluid, which has a high surface tension, it's very watery, that keeps the lungs stuck, so to speak, or adhesed to the thoracic wall. And that has consequences for the mechanical ventilation function of the lungs. Okay, so let's go into ventilation. This, by the way, is also covered in other lectures. And so I would recommend using this part of my lecture as ancillary material to Dr. Zumwalt's lecture. So because the lungs are stuck to the thoracic wall and the diaphragm, that means that those structures can help expand the lungs and also deflate the lungs. So the diaphragm is the main respiratory muscle. and it is located here. It divides the thoracic cavity from the abdominal cavity. Now, also on this diagram, you can see approximately where the lungs would sit. Here's where the right lung would be located in the thoracic cavity, and here's where the left lung would be located in the thoracic cavity. And it is separated by the heart, which is located here. So we take the thoracic cavity and we divide it actually into 3 subsections, and that is why the lungs are located in the pleural cavity, and the heart is located in the mediastinum, or the cardial cavity. We typically call it the mediastinum. Anyway, the lungs are laying on top of the diaphragm, and so when the diaphragm contracts, that allows the lungs to expand and take in air. Now, as you can see, there are lots of other muscles associated with the thoracic cage. There are the intercostal muscles, and here we see the innermost intercostal muscles. There are the internal intercostal muscles here and the external intercostal muscles here. And then we have these other muscles, the first set of which I'm showing here, called the scalenes. These are muscles that are attached to the ribs, but they are a little bit more involved in moving the ribs themselves. So the diaphragm is what's called a passive muscle, in the sense that when the diaphragm contracts, it mediates the passive inhalation of air. But if I want to mediate active or forced inspiration of air, I'm going to recruit muscles other than the diaphragm that are attached to the rib cage. So any other muscle attached to the rib cage that allows the rib cage to lift, then those are going to be active inspiration muscles. So those include the scalenes, and those would include the intercostals. Indeed, the external intercostal is an active muscle, or it's a muscle for active inspiration or forced inspiration, as are the scalenes. So here are some examples of others. The pectoralis major and minor also attach to the rib cage. And so when I contract those muscles, if I fix the arms in place, then I can also lift the rib cage. So those are active muscles, or active muscles of respiration, or muscles of forced inspiration. There's, as you can see, lots of different nomenclature for this. Another muscle is the sternocleidomastoid. It attaches onto the clavicle and the manubrium, and so it can also lift the rib cage when necessary. So here is an example of what it looks like to cause pulmonary ventilation. Again, anything that expands the rib cage or increases the thoracic cavity will mediate inhalation or inspiration. So we go here first to the diaphragm. The diaphragm, when it contracts inferiorly, which expands the pulmonary cavities on either side, and that allows the lungs to take in air. Or we can use the external intercostals, which will expand the rib cage anteriorly and laterally. We can use any of the other big muscles that I mentioned. Those will increase the thoracic volume. By contrast, anything that decreases the thoracic volume, either passively or actively, will mediate exhalation. So when the diaphragm relaxes, it moves up, which reduces the pulmonary volume and allows air from the lungs to go out. Or if I'm using my internal intercostals, that allows the rib cage to swing down. Okay, so I think now we're ready to move on to heart. And so we'll talk about the heart and mediastinum first. So just to reiterate the idea of the different cavities, the location of the heart is in the middle of the thoracic cavity. It is in an area called the middle mediastinum. Mediastinum basically refers to the middle part of the thoracic cavity. The pleural cavities would be on either side. So the right lung would be located here, and the left lung would be located here. Notice that all 3 structures, the right lung, the left lung, and the heart, all sit on the diaphragm. So when the diaphragm moves up and down, the lungs move up and down, but so does the heart. it also moves up and down. The difference, of course, is that the heart does not have alveoli. So the location of the heart is important. The apex of the heart points towards the fifth intercostal space on the left side. And it has because of that, you can think of the heart as a pyramid that I have tipped anteriorly and to the left. So again, here's the apex of my pyramid. I tip it forward and to the left, and that is the orientation in the thoracic cavity. The consequence of that is that the right ventricle is located anteriorly. And that's important. Right ventricle is always anterior. It's right behind the sternum. The left atrium, or the base of the heart, is located posteriorly, right up against the vertebral bodies. So by default, the right atrium is located on the right border of the heart, and the left ventricle is located on the left border of the heart. So just as a review, the right ventricle is in front, the left atrium is in back, the right atrium is to the right, and the left ventricle is to the left. Another thing to keep in mind is that the inferior surface of the heart, which is comprised of both right and left ventricles, is also called the diaphragmatic surface of the heart, and it sits, of course, on the diaphragm as a result. Okay, so if I took a cross section through the middle of the heart to show this four-chambered view, the question that I have for you is, which chamber is what? And so pause the video and take a moment to try to figure out which chamber is what based on the orientation that I just gave you. One of the things to keep in mind is that in radiological convention, and this is a CT scan, in radiological convention, the left side of the image, located here where my pointer is the right side of the body. And the right side of the image is the left side of the body. The top part of the image is the anterior part of the body, and the bottom part of the image is the posterior part of the body. Okay, so if the top part of the image is the anterior part of the body, and the most anterior chamber is the right ventricle, then that means B is the right ventricle. If the vertebral body shown here in cross-section is on the posterior side of the body, then that means the chamber closest to it, D, would be the left atrium. And now remember, in radiological convention, right is left and left is right. So that makes C the right atrium and A the left ventricle. Just for fun, we're going to talk about some other structures later in this lecture, but you can see a little thin wisp right here and right here. This is the valve between the left atrium and left ventricle, so that would be the mitral valve. And then we see another little thin wisp right here, and that is the valve between the right atrium and the right ventricle, so that would be the tricuspid valve. And this structure here, which is my favorite structure ever, is called the moderator band. And we'll talk about the moderator band in a bit, but as you can see, it extends from the interventricular septum located here. Okay, so let's talk about blood flow. Blood enters into the right atrium. We'll talk about it in series first. The blood enters into the right atrium from the superior vena cava, the inferior vena cava, and not shown here is the coronary sinus. That is the drainage for the heart. Blood then flows from the right atrium into the right ventricle, located here at number 2, and then from the right ventricle to the lungs. So the valves that blood passes from 1 to 2, or from the right atrium to the right ventricle, is the tricuspid valve, and the valve through which the blood flows from the right ventricle to the pulmonic or pulmonary trunk is the pulmonic valve or pulmonary valve. Blood branches to the right and left pulmonary arteries, goes to the lungs, and then it returns via the pulmonary veins. And the pulmonary veins, there are 4 of them, two from each lung, will enter into the left atrium, which is located here. And then from the left atrium, blood flows into the left ventricle, then out the aorta. So the valves through which blood flow from the left atrium to the left ventricle is the mitral or bicuspid valve. and the blood that flows into the aorta gets there through the aortic valve. So this causes two circuits. Oh, I should finish by saying the blood flows from the aorta to the rest of the body, whether it's the upper part of the body or lower part of the body, and then the blood returns. So there are two circuits as a result of this blood flow. The pulmonary circuit is the circuit that is receiving deoxygenated blood and returning oxygenated blood. And the systemic circuit is receiving oxygenated blood and returning deoxygenated blood. An easier way to remember this is that arteries, regardless of what kind of blood they carry, are vessels that move away from the heart, and veins are vessels that move toward the heart. Just for a little trivia, on average, your heart beats 75 times a minute, which is 108,000 times per day. And the average cardiac output is 5.25 liters per minute. This guy is working really hard. I went through the blood flow in series, but in reality, blood flow is happening in parallel. So blood flows into the two atria at the same time during atrial diastole, and then from the atria into the ventricles during ventricular diastole, and then from the ventricles out to the body or to the lungs during ventricular systole. So it's a parallel circuit, and we often consider the heart as a right heart pump and a left heart pump that are working together but separately. Okay, so these are the structures involved with blood flow through the heart, which we just covered. And so we're just going to review that quickly. Blood flows into the right atrium via the inferior vena cava or the superior vena cava, or the coronary sinus, which isn't shown here. So here's the right atrium. Blood flows past the tricuspid valve into the right ventricle, and from the right ventricle, it flows into the pulmonary trunk through the pulmonary valve or pulmonic valve into the lungs, and then blood returns via the pulmonary veins into the left atrium. It flows into the left ventricle through the mitral valve, and then from the left ventricle out to the aorta via the aortic valve, which you can barely see right here. So there are some other structures that are important for all of this to work, namely the leaflets of the atrioventricular valves. So generally speaking, the atrioventricular valves are the tricuspid and the mitral, because they are located in the atrioventricular border between the atrium and the ventricles. So each of these valves has these flappy leaflets. The tricuspid valve has 3 of these leaves, and the mitral valve has 2. And so for each leaf, you typically have 1 papillary muscle, which are located here. And the papillary muscles tether to the leaves via these structures called chordae tendineae. So we can see 3 papillary muscles here on the right, because we have 3 leaflets for that valve, and then 2 papillary muscles on the left, because we have 2 leaflets for that valve. And then here again is the moderator band. Again, my favorite structure in the body, so we'll talk about that in a bit. As far as the aortic and the pulmonic valves, these are both tricuspid semilunar valves. And when the semilunar refers to the shape of the cusp, and so there's 3 of these. I only have 2 hands. There's 3 of these in each of those valves. Now these, as you can see, are not tethered down by chordae tendineae or any cords at all. They are actually more like doors that open, and then when they close, there's a little nodule that's located right here on each of the different cusps that allow these valves to close without actually prolapsing back into the ventricles. Some other things to note is that the right ventricle wall is relatively thinner than the left ventricle wall, and that there's lots of reasons for that, which you'll go over in physiology. Okay, so now let's transition into talking about the pericardium, which again has the same structural principles as the pleura. What we see here, so we'll start with the pleura actually. Here's the right lung and here is the left lung. And you can see the parietal pleura up against the ribs. Parietal pleura actually has 3 different names based on its location. Up against the ribs, we call that specifically costal pleura. And then we have pleura that is next to the mediastinum. That parietal pleura is called mediastinal pleura. And then we have parietal pleura that's on the diaphragm, and that pleura is called diaphragmatic pleura. The visceral pleura, which is shiny, drawn here in the lungs, does not have any specific names. It's just called visceral pleura. Okay, so that's the lungs. So what's happening with the heart? Well, the heart also has a serosal membrane. It has a serosal membrane on top of it, or on its surface. And of course, because it's on the organ, we're going to call that a visceral pericardium. So here the serosa name is pericardium, whereas in the lung, the serosa name was pleura. So the heart has a visceral pericardium on its surface. Then that means it also has to have a parietal pericardium up against what? Where is our body wall located here? The lungs have the body wall as the thoracic cage, but the heart needs to have its own body wall. So let's take a look at that. So if we take the heart out, what we are seeing here is the parietal layer, or the parietal pericardium, because the heart is gone, so the visceral pericardium goes with it. This is the parietal pericardium, and it is lining a body wall structure called the fibrous pericardium. So this is the only place in the body where the body wall is actually splitting a cavity. And it does this because of embryological reasons. But one of the ways we can prove that to ourselves is that the phrenic nerve, which travels here, runs on top of, or just external to, the fibrous pericardium. And the phrenic nerve is a somatic structure. So all body wall structures are somatic, and all somatic nerves are voluntary. So how do you know? Well, hold your breath, and you just proved it to yourself. You can control that. So let's take a look at this in a little bit more detail, because we have a cutaway section of this that helps us understand this. So let's talk about it here. So here is our heart surrounded by its fibrous pericardium. So if I take a cutaway of, let's say, this slice here on the left side of the heart, this is what I'd see. So I would see the heart wall, which is designated here by myocardium, the serosal layer on the surface of the heart, which is this. So because it's on an organ, we call it visceral. Because it's a serosa surrounding the heart, we call it pericardium. So this is visceral pericardium. Or its long name is visceral serous pericardium, but we already knew it was a serous layer. So visceral serous pericardium. Histologically speaking, it's the same layer that we consider as the epicardium. It's the same thing, right? On the other side of this pericardial cavity, which I'll go over in a second, is our body wall. And a body wall in this case is the fibrous pericardium. If this were the lung, this would be the thoracic cage, right? But here we're talking about a different organ. So fibrous pericardium. That fibrous pericardium is inside world, and it doesn't want to be exposed to the pericardial cavity. So it has to have a layer, or a serosa, that lines it. And that is our parietal pericardium, or parietal serous pericardium. So just like with the lungs, we have a space between the parietal serous pericardium and the visceral serous pericardium. And that space is called the pericardial cavity. So pericardial cavity is to the heart, as pleural cavity, or pleural space, as I showed you, is to the lungs. So we're missing a layer here, aren't we? Because our fibrous pericardium is inside world for the heart, but I've got the lungs here, which would be located where all the words are. And we don't want the fibrous pericardium to be exposed to the lungs. So yes, there is another layer that is located just laterally to the fibrous pericardium, and that's the pleura. That's the parietal pleura, or the mediastinal pleura, which I referred to earlier. Now, the pericardial cavity and the pleural space are different in one major respect. The pleural space is a potential space. And the reason why we call it potential space is because in vivo it doesn't really exist. And why doesn't it exist? It doesn't exist because it's filled with serosal fluid. So the lung adheres to the thoracic or diaphragmatic wall, if you will. It's adheres like this, and the serosal fluid is what keeps those two layers stuck together. We don't want that to happen in the heart. So the pericardial cavity is a true space. It's not filled with enough fluid that would cause surface tension between the visceral serous pericardium and the parietal serous pericardium. So that pericardial cavity needs to be empty. It's filled, there is a serosal fluid in there, but it's mostly filled with air. The problem becomes when the pericardial cavity is filled with too much serous fluid, or in trauma cases with blood. The pericardial cavity is the potential is the space, excuse me, it's not a potential space, it is a space. The pericardial cavity is the space into which the heart expands during ventricular diastole to fill with blood. If that cavity is filled with too much fluid, then the ventricles cannot fully expand to fill with blood, which means it causes a cascade of issues with the filling of the ventricles, which you'll learn more in physiology. But the bottom line is the heart figures out it can't fill with as much blood, which means it can't eject as much blood, which means it has to work harder to do the same amount of work that it was designated to do. Okay, so let's take a look at the heart wall. Now, by way of orientation, this is the parietal serous pericardium, which is drawn in or colored in purple. And this, what looks like a fibrous layer, is indeed the fibrous pericardium. Okay, and then on the heart side is the epicardium. Histologically, which you might remember has the same name as the visceral serous pericardium. The heart wall itself is comprised of myocardium, cardiac myocytes, and then the chamber, if we're talking about, this looks like the left ventricle as it's drawn here. The chamber over here is lined by its own epithelium, and it is called the endocardium. So what's super interesting here is remember that you have to have an epithelium every time you're separating the inside world, in this case the myocardium, from the outside world. So yes, the cavity spaces in the heart are what I would consider as outside world, if you will. So now let's talk about heart chambers and valves. So I'm going to run through this section relatively quickly because I'm going to post another video for you that goes into this in a little bit more detail. And all of the images are labeled. So the right atrium is labeled here as 1 on the right auricle. The auricle itself is not the entire right atrium, but it does designate where the right atrium would be located, which of course here in anatomical position is the right border of the heart. 2 is the right ventricle, and remember it's the most anterior of the chambers, right behind the, or right posterior to, or just posterior to the sternum. The left atrium is designated here by the left auricle, and as you can see on the inferior surface of the heart, it's a large chamber. So it's not just that little auricle, it's quite big. In fact, actually, you can see the right atrium also in its full size. Number 4 is going to be the left ventricle. Notice that it does form the left border of the heart, but most of its surface is on the inferior border of the heart. So if we open up the right atrium, there are a number of structures to note. The first is the superior vena cava, labeled 1, and the inferior vena cava, labeled 2, entry into the right atrium. Finally, we can see the coronary sinus, here labeled as number 3. So blood is flowing into the right atrium via these 3 structures. Number 4 is the interatrial septum, and there is often a visible impression called the fossa ovalis on the interatrial septum. Sometimes it's a hole, in which case it would be called a patent foramen ovale. This is the embryological shunt from the right atrium to the left atrium. The atrium wall, as you can see, has a smooth part and a rough part. The rough part is called the pectinate muscles, labeled 6. The smooth-walled part doesn't really have a name other than the smooth-walled part. And then if we look at the cardiac conducting regions, 7 locates where the sinoatrial node is typically located, and 8 is where the atrioventricular node is typically located. These structures are, or I should say these numbers designate areas. They don't designate actual structures. So the SA node and the AV node are cardiac conducting cells. They're not nerves, they're not ganglia, but they are cardiomyocytes that have been modified to transmit electrical signal, receive and transmit electrical signal. Number 9 is the tricuspid valve, which is the valve that is the doorway for the right atrioventricular orifice. So let's open up the left ventricle. And what we see here is the blood will flow out of the right ventricle via the pulmonary trunk, which is number 1. But I'm actually going to start with the tricuspid valve since that's where blood is flowing, or that's the route blood is flowing. So blood would flow from the right atrium through the tricuspid valve. And as you can see, the valve leaflets here are tethered to the papillary muscles in 3 via the chordae tendineae, which is located in 4. Number 2 are the trabeculae carneae. They are a thicker trabeculated structure than the actual pectinate muscles, but they look chorded in the same way. Number 6 is the moderator band, and actually it looks like this number 6 floated away. Number 6 should be located on this structure here. I'm going to highlight it. I apologize for that. So here is the moderator band. 6 should be right here, and the moderator band extends from here to there, from the interventricular septum. Okay. Number 5 is the pulmonic valve. Notice that it is also a semilunar valve, 3 cusps. All right, and then blood flows out through the pulmonary trunk. Here is the left atrium. It receives blood from the pulmonary veins, which are not shown here, but blood flows into this region. And then it is bordered from the left ventricle via the left atrioventricular orifice and the mitral valve, which is shown here in number 1. So blood flows from the left atrium into the left ventricle. So here's the view of the left ventricle, which is a little more detailed in a cross section down in the lower left. So notice the thick heart wall. Notice our papillary muscles, which tether the mitral valve, and blood flows from the left ventricle into the aorta via the aortic valve, which is shown here, number 2, and then into the arch of the aorta. Okay, so now we're going to talk about the heart valves. So in this drawing, we've taken a slice through this plane, through the plane of the atrioventricular orifices, and through, you'll notice that the valves are located all in that same plane. So what to note here is that in the drawing, the anterior surface of the heart is at the bottom and the posterior surface of the heart is at the top. And it's just an unfortunate orientation for this particular drawing because typically we would look at it the other way. But to note is the arrangement of the valves. So because the right ventricle is the chamber in the most anterior portion of the heart, the pulmonary semilunar valve would, of course, be the most anterior of the valves. And here it is here. Then going posteriorly, we have the aortic semilunar valve. And then on the right side of the heart is the right atrioventricular valve, and on the left side is the left atrioventricular valve, again, also known as the tricuspid and mitral valves. Something to note that is really cool here is you can see the little ostia or holes in the aortic semilunar valve, which correspond to the left coronary artery and the right coronary artery. So that's pretty cool. Okay, this is a really important structure of the heart. It's called the fibrous skeleton. And actually, let me go back to the previous slide. The fibrous skeleton is a structure that surrounds each of the different valves. Okay, so if we draw it this way, it's drawn here in this tan color or orange, or I don't know what color that is. So, but you can see that it's surrounding all the valves. This fibrous skeleton forms the scaffold for the valves. And it kind of looks like a drone to me, actually. So it it holds the valves in place and on the same plane. But the fibrous skeleton actually has a much more important function. And that is that because it is comprised of non-cardiac conducting cells, in other words, it's electrically resistant, then that means electrical signal that travels from the atria can't get to the ventricles via lateral wall pathways. The only place in the fibrous skeleton that is not electrically resistant is this little teardrop looking drawing here, and this is the location of the atrioventricular node, or the AB node. And so that is super cool, that the electrical conductance from the SA node going around the atria focuses in on the AB node. If we didn't have the cardiac skeleton or the fibrous skeleton, then, well, we wouldn't be here, because the chamber walls would not contract in the correct way to be able to push blood to the right direction. So again, here are the atrioventricular valves. They're surrounded by that cardiac skeleton. In this drawing, the atrioventricular valves are open, which means blood is flowing through them. So this stage of blood flow is ventricular diastole. Here is an image when the valves are closed. So this would be a situation during ventricular systole. So why do the valves close during ventricular systole? Well, we don't want blood flowing back into the atria. We want the blood flowing into the aortic and pulmonic tracts. So diastole is when the atrioventricular valves are open, and systole is when the atrioventricular valves is closed. So here is the opposite view. So here now the atrioventricular valves are closed and the aortic and pulmonic valves are open, which means this is systole. Now this valve here is the aortic valve, and I know that for two reasons. One, I have my little holes for my coronary vessels. Number two, here is the mitral valve, and it is, you can see that this leaflet located here is intimately connected with the aortic valve. So that is a clinically important structural feature because it has consequences for valve replacement. So if I replace the mitral valve, then I have to be careful about whether I need to replace the aortic valve or not. So because if I remove this valve, then it's going to affect the cusps on the aortic side. So I'm going to go back a slide. So again, my atrioventricular valves, when they're open, that is during ventricular diastole, because blood is flowing from the atria into the ventricles. The ventricles are filling. During ventricular systole, my atrioventricular valves are closed and my aortic and pulmonic valves are open. Okay, so now we're going to talk about my favorite part of the heart, which is the cardiac cycle. All right, so the heart receives sympathetic and parasympathetic innervation. The parasympathetic innervation emerges from the medulla via the vagus nerve, and its job is to decrease heart rate. So that's what this line shows here. The ganglia that are located here, represented by this synapse, are located in a plexus on the aorta right here, called the cardiac plexus. It is part of the cardiac pulmonary plexus. The pulmonary part, of course, is going to the lungs. The sympathetic nervous system is shown here. Its job is to increase heart rate and the force of the contraction. Notice that there are no ganglia drawn into this image here. The cell bodies from which these nerves emanated are located in the neck, specifically, but not exclusive to, the sympathetic cervical ganglia, in particular the stellate ganglion. So in summary, the vagus nerve drawing here is a preganglionic neuron and its axon, and then these are postganglionic, and the entire sympathetic tree here that you see in this drawing is all postganglionic. Okay, so one of the things that the sympathetic nervous system does specifically is to innervate the sinoatrial node. It also innervates the atrioventricular node, to be able to increase the rate of contraction. But another thing to keep in mind is that the parasympathetic nervous system has a large amount of innervation to the atrioventricular node. And what that does is it slows down the conduction through the heart. So that way the SA node serves as the pacemaker and the AV node serves as the gatekeeper. So here are those nodes specifically. So here's the SA node. Again, it receives largely sympathetic innervation. Here's the AV node. It receives largely parasympathetic innervation with some sympathetic innervation. And of course, the sinoatrial node will receive some parasympathetic, but again, this is the difference in balance. So, okay, this shows a nerve pathway, but the thing to keep in mind is that these are not nerves. The cardiac conducting myocytes are cells that have been modified to be able to transmit electrical signal. We just draw them as nerves, but they're not nerves. So please keep that in mind. So when the SA node fires, it sends its chain of electrical stimulation throughout the atria in 3 dimensions, as if it's like water pouring over a beach ball, all right? And so that electrical stimulation goes around the atria and causes the atria to contract during atrial systole. But remember, we have the cardiac skeleton, which is right here in the middle, that tethers the atrioventricular valves and the semilunar valves. And the only place in that cardiac skeleton that is not electrically resistant is where the AV node is located. So when an electrical signal travels down, say, this internodal pathway, it cannot extend down the lateral walls of the ventricles on either side. And that is huge, because that means that the atria contract and they push blood into the ventricles. If the ventricles contracted from the side, they would also push blood towards the apex of the heart, which is not where we want blood to go. We want blood to go up or superiorly through the aortic valve or the pulmonic valve. So how does that happen? So our SA node fires, we get the electrical signal which coalesces or converges on the atrioventricular node. It fires, assuming that we have a typical parasympathetic tone that doesn't prevent it from firing. Then the electrical signal travels down the bundle of His here, which is in the interventricular septum. And then the bundle of His will branch into the right and the left bundle branches. So the electrical signal travels down the right and left bundle branches, down to the Purkinje fibers, and back up superiorly on the heart wall. And when you study EKG, you'll be able to actually see how that works electrically. But the anatomical effect is that my interventricular septum will contract, and the ventricular chambers will contract from the apex superiorly, pushing the blood up towards the semilunar valves. Okay, so that was a quick primer on the cardiac cycle, which is very cool. All right, so I brought up the moderator band earlier. Why is it so important? The moderator band is a part of the interventricular septum muscle wall that branches to go to the papillary muscles. And it's not shown in this diagram, but it's represented basically by these branches here in the drawing. Essentially, the moderator band acts as a shortcut for electrical stimulation to go from the septum to the papillary muscles before the Purkinje fibers see that stimulation. Why is that important? In the right ventricle, the blood flow going to it from the right atrium travels into the right ventricle, but then when the ventricle contracts, that blood will go to the path of least resistance. It can either go out the pulmonic tract, or it can go back into the right atrium. Well, we don't want blood regurgitating into the right atrium. So we anticipate the force of blood flow going back into the right atrium by getting the papillary muscles to start contracting just before the rest of the ventricle contracts. The analogy I like to think about is a person at the beach that is flying a kite. Imagine if you could anticipate a gust of wind, right? And so me as the person is the papillary muscle, the string is the chordae tendineae, and the kite are the leaflets of the tricuspid valve. So if I start pulling down just before the wind starts to hit the kite, I can prevent my kite from either flapping away or tearing away. And that's the idea here. What's interesting about this is we don't have a moderator band on the left side. And that is because the pressure differences between the right atrium and the left atrium are slightly different. So the right atrium has slightly less pressure than the left atrium does. And so blood coming from the right ventricle will tend to flow back into the right atrium if we don't have the tricuspid valve tethered down. But in the left ventricle, blood tends not to go back into the left atrium because the pressure is high enough in the chamber to keep the valves from prolapsing. I think that is a super cool concept and modification. Okay, let's talk about coronary circulation. I have another video on this that goes into this in a lot more detail. So what I'm going to cover here are the pairs of the vessels. And then I'm going to refer you to my other video that goes into more detail on how this works. The bottom line is that the heart is perfused with oxygenated blood during diastole. So the ventricles contract, blood flows out of the aortic tract, if we're talking about aorta, into the arch of the aorta, the ascending aorta and the descending aorta. During diastole, some of the blood didn't get quite passed over the arch. It's sort of like the roller coaster car that didn't quite make it over the hump of the hill, right? And starts to now go backwards, which never happens. But in this case with blood, it works. So the blood that didn't quite get over the arch flows backwards towards the aortic semilunar valve and actually will flow into the coronary vessels themselves while the heart itself is filling with blood, or the ventricles are filling with blood. Okay, so the pairs of the coronary vessels include the right coronary artery here, and it travels with the small cardiac vein. The left coronary artery is here, but I'm actually going to continue and talk about the right coronary artery first, then we'll talk about the left coronary artery. So here's the right coronary artery. It swings around, and it comes around the atrioventricular sulcus, and typically in a right dominant heart, it will branch to give the posterior descending artery or the posterior interventricular artery. And the vein that travels with the posterior descending artery is the middle cardiac vein. So just as a quick review, right coronary artery travels with the small cardiac vein, and as it swings around the atrioventricular sulcus, it branches as the posterior descending artery, and that artery travels with the middle cardiac vein. The posterior descending artery supplies the posterior 1/3 of the interventricular septum, which is clinically important. But let's talk about the left coronary artery. Actually, before that, I should mention that there's a marginal artery that is branching off the right coronary on the right surface where the heart is nearest to the diaphragm. Okay, so the left coronary artery, I just really want to get to the left coronary artery. So the left coronary artery is here, and it's pretty short, actually. It's much shorter than what's drawn here. It branches immediately into the left anterior descending artery, or the anterior interventricular artery, which needs to be corrected here on the words. The anterior interventricular artery is also known as the left anterior descending artery. So please correct that in your notes. So the left anterior descending artery is accompanied by the great cardiac vein, which is shown here. The other branch of the left coronary is the circumflex, and you can see it going underneath the auricle right here. And it's barely visible going around the left atrium on this side. But the circumflex artery travels around the left side of the heart in the atrioventricular sulcus, and it is also accompanied by the great cardiac vein. So that means that the left anterior descending and the circumflex are both paired with the great cardiac vein. All right, the left anterior descending artery supplies the anterior 2/3 of the interventricular septum. All right, let's finish off by talking about pulmonary and cardiac auscultation. So here's a picture of pulmonary auscultation. And what this shows is the general zigzag pattern that a clinician could use to listen to the different lobes of the lung. The lungs are easiest auscultated on the posterior side or from the back. And the main reason for that is because the heart is so loud, it gets in the way in the front. It doesn't mean you can't hear for the lung sounds from the front, it just makes it a little bit harder. What I do want you to note is that from the front on 1 and 2, you can listen for the lungs, particularly the cupola, right here, just superior to the clavicle. And that's an important auscultation site. As for the heart, the principle to keep in mind is that we're going to listen for heart sounds where blood is flowing towards you. So if I want to listen for the aortic valve, for instance, which is shown here in green, I want to use the second intercostal space on the right side of the sternum because blood is flowing in that direction through the aortic valve. I'm going to take advantage of the Doppler shift, which gives me a louder sound and a higher frequency so I can hear the sound better. If I want to listen to the pulmonary valve, I'm going to listen at the second intercostal space on the left side because that is the direction in which blood is flowing through the pulmonary valve. And then by the same convention, if I want to listen for the mitral valve, I listen at the 5th intercostal space at the midclavicular line because blood is flowing through the valve in that direction. And then if I want to listen for the tricuspid valve, I listen at the 5th intercostal space on the left, just next to the sternum, because again, blood is flowing in that direction. The table actually shows this really nicely and is a really good summary of cardiac auscultation and where you would hear those sounds. All right, well, that is it for this lecture. Thanks for paying attention, and we will see you in lab. Have a great day. Bye.